You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I'd like to welcome my guest, Eleftherios Mylonakis. He's the Charles C.J. Carpenter, MD, Professor of Infectious Diseases, Professor of Medicine, Assistant Dean of Emergency Medicine, and Professor of Molecular Microbiology and Immunology at Brown University. We're going to be talking today about his study on microbial pathogenesis and host responses. It's an excellent call we have ahead of us, and Eleftherios is working on how to deal with antibiotic-resistant bacteria that can kill people, such as MRSA. So it's a very important area that he works in. Antibiotics, there's very few new ones coming, if any, and antibiotic resistance has been rising in bacteria a lot because of the use of antibiotics over the past few years. So this is a critical thing that he's working on. So I'd like to welcome him. Thank you.
You're very welcome. I'm with the Department of Medicine, not emergency medicine. So we're dealing so much with emergencies. No problem.
So what's the focus of your work currently? What are you working on most recently?
Our last decade has mostly been on infectious diseases and drug discovery. So that is what we spent most of our time investigating.
When you say infectious diseases, are these diseases that happen in the first world or like third world countries? Where do they happen and what are they?
Yes. So, Rich, our main interest has to do with resistant bacteria, and especially this pathogen called MRSA, which is Methicillin-resistant Staphylococcus aureus. And unfortunately, this is now widespread. It happens in every country and in the U.S. as well as the rest of the world, and even including developing countries. So this has become very widespread and a real problem for patients and for physicians to treat.
Yeah, I thought Staphylococcus aureus is a very common microbe that exists in us on a beneficial basis. Is this a certain strain, or what have you identified? How does it become pathogenic?
Well, yes, Staphylococci are very common, and they live on our skin mostly, and they're part of what we call a microbiome of the skin overall. However, that is a particular group of Staphylococci that is called coagulase negative, and especially, obviously, Staphylococcus epidermidis. Epidermis is the skin. Epidermidis is the type of Staphylococci that are not very pathogenic, and they're part of our skin flora. However, Staphylococcus aureus also can colonize individuals, but has a lot of virulence factors that make it very keen to cause infections. And what happens On top of that is that it progressively developed resistance to a particular class of antibiotics, the penicillin-like antibiotics like methicillin, and that is what became MRSA, which is the methicillin-resistant type of Staph aureus, which Staph aureus is the pathogenic Staphylococcus. The interesting part about Staphylococcus and what makes it very concerning is exactly what you said, Rich, that it is very widespread and can colonize. So it can live on our skin, it can live and can spread from person to person. So for the most part, 1 out of 3 cases of this resistant bacteria that come to the hospital with severe infection, 1 out of 3 patients come from the outside. They are not even hospitalized at the time of their diagnosis. I remember one young woman that came with a very severe infection from MRSA, and she was just 20 years old. She had never been admitted to the hospital, and she came and she was septic, and she had to go to the ICU. And unfortunately, this is very common. And there are some populations that are even more prone to get MRSA infections. But essentially, most everybody can develop an infection like that.
Where does this particular type of bacteria exist when it's not causing harm? Where does it exist on us? Is it in the skin?
It is in the skin. It is especially likes the environment of the nose, the outside 1/3 of the nose. It's a place where we usually try to screen when patients get admitted to the hospital. We try to see if they carry this type of bacteria. So that's the place where we screen. And the majority of people that are colonized with MRSA, or other Staph aureus, they are going to give us a positive test when we test a swab from their nose. But it can be on our skin, it can be on the armpits, it can even colonize some abiotic surfaces. So that makes the decolonization, trying to prevent infection, trying to prevent infection and take away the colonization, very difficult, because it often fails due to the widespread of this bacteria.
So what one theory, again, it's my theory, but if they're living in, essentially homeostasis, or they're happy in all these places on our skin and other surfaces, what is keeping them in check? What is keeping them happy? Is it other microbes? Is it availability of food? Why do they get to a state where they go crazy?
Well, there is an easy answer there, and there is a more in-depth answer. So I'm going to give you the easy answer first, and then we'll try to venture into a little more on the complexity of the issue. So the easy answer is that if they're on our skin or in our mucosal surfaces, the skin and the other barriers that we have against the pathogens work very well. It prevents the Staphylococcus from entering our bloodstream. It prevents the Staphylococcus from spreading to our heart valves, to our bones, to cause an abscess. So it keeps it in check just by the fact that there are barriers that exist and they're part of our body. Now, and then once we have a simple trauma, like a scratch, or if an IV drug user uses a needle that is colonized with MRSA, or we are in a motor vehicle accident, for example, where we have even a minor break into our barriers, then the Staphylococcus will penetrate and will start spreading through the bloodstream and become pathogenic just because it broke through this main barrier. Now, this is the easy part, as I said before, the easy part of the answer to your question. The more complex part has to do with the increased understanding that we have for that not everybody is equally susceptible to pathogens. So it's not only the barriers, but it's also the host responses, the previous exposure of the host to the pathogen, and also the overall immune status of the individual. People that who are immunosuppressed, for example, people that who are neutropenic, individuals that are at the extremes of extreme age groups, certainly are more prone to develop more severe infections. Nevertheless, this, and also there is person-to-person variability. We still haven't understood exactly the different components of that, but not everybody responds the same way to the same pathogen.
Do people have MRSA on their skin and in their nose, or they have a different strain that is not MRSA? Like, how can you even tell that a given Staphylococcus is resistant or not? Can you tell by the surface coat it has, or just by its behavior, or you sequence it? Like, how do you know?
Yeah, so this is something that we do routinely. Overall, you should keep in mind that a number of different categories of individuals, they have a significant colonization percentage with MRSA. And for example, athletes, especially collegial athletes, they are at risk of becoming colonized with MRSA. Patients in the ICU, individuals that have required dialysis because of renal failure, of course, they can have all those categories. They can have up to 10% or more of the individuals that belong to these categories can be colonized with MRSA. So this is just to give you a frame of how common MRSA colonization can be. Now, how do we test? How do we know if it's MRSA or not? Well, as I mentioned before, we routinely screen patients that get admitted to the hospital, especially those that get admitted to the intensive care unit for colonization with MRSA. And we used to do a simple culture and grow them, but now there are specific media, and also there is a molecular assay, PCR-based assay, that quickly gives us a result if the person is colonized with MRSA or not. If the person is colonized with MRSA, we have to take some particular precautions in order to try and prevent spread of MRSA to others. And we have to wear gloves. We have to probably use a separate room just for this patient, etc.
So if you swabbed a healthy person, would you expect to find any MRSA or no?
It depends on the category, but it can happen. As I said before, collegial athletes are colonized overall 10% or more. Actually, collegial athletes have higher rates of colonization with MRSA than patients in the ICUs, which is remarkable. Non-collegial athletes, pro athletes also, I'm sure that you have heard every so often, every couple of years, there is a professional team that has to close down their facilities and fight MRSA and try to decolonize. And there are well-known athletes that have come out, have become have gone public with their stories of MRSA infection that complicated their surgeries and operations and procedures. So even the healthy people, the athletes, the young athletes in particular, they are also very keen to become colonized, because colonization is part of living in a community, essentially, of other people. So it's not that you need to have an immune system that has a defect necessarily in order to become colonized. And it's unfortunately quite common.
But what if instead looking at it as a pathogen, it's just a commensal organism, and it's it's mutualistic with us for the most part, but then conditions occur under which it proliferates, like C. difficile in your guts. If you look at it in that respect, then maybe perhaps there are other microbes or other things in addition to the immune system that are also commensal with us that are keeping it in check. And if you looked at, let's say, the entire microbial constituent around MRSA, when in a healthy person versus a sick person, perhaps you'd see like other species of bacteria that were there or not there under different conditions that MRSA was in. Infectious stage, non-infectious stage. It's just another way to think about it. Maybe, I don't know, maybe it's no good.
Yeah, there is no question about it. There is no question that your thinking is on the right track. Indeed, it's very reasonable to assume that there is an interplay in the colonization status between the MRSA bacteria and the microbiome that exists around it. I don't have any hard data to tell you, because this is something that we want to study. What is the interplay between the host responses, the MRSA, and the microbiome around it? Because you're looking at a situation where you don't know the causality. Maybe you can say one step back, maybe the microbiome of the individual get disrupted, the skin microbiome, for example, or the microbiome in the nose gets disrupted first and makes the individual more prone to develop colonization with MRSA also. So your thinking one step even further, you can even assume that there is some causality between the disruption of the local microbiome and the development of the colonization. However, those studies, we are looking into those. What I can contribute to your thinking and to your question about it is a series of studies that we did just a month or so ago, where we took MRSA isolates from people that didn't have an MRSA infection, they were just colonized with. And we found that the virulence traits and the resistant traits of those isolates that they just called colonization, they were identical to those from isolates from bloodstream infections. So the MRSA bacteria that colonize us seem to be as virulent as those that cause infection. Further substantiating your argument that those bacteria are very pathogenic, something must be controlling them beyond the barriers of the skin that keep them outside of our body.
Yeah, I would think that it just sounds to me very similar to C. difficile. It's always there. And then when a dysbiosis is caused by antibiotics, for instance, it allows C. difficile to proliferate because maybe its competitors, it's stuff that's keeping it in check, is dying off. So it sounds like perhaps the same thing happens. Maybe the mechanism even is due to antibiotics. Maybe it's affecting also the skin microbiome, and maybe it's killing off the gatekeepers that keep MRSA in check and allow it to proliferate.
Yep, there is a similarity that I see there on this argument. Indeed, we usually say that about 1 out of 20 individuals is colonized with C. diff, and indeed, the colonization exists as part of how C. diff gets spread. However, there is one difference here. C. diff does not need to penetrate the mucosal barrier in order to cause infection. So just a disruption from the use of broad-spectrum antibiotics is enough when the rest of the microbiome is completely disrupted for C. diff to cause infection. For Staphylococcus aureus, the microbiome around it might keep it in check, but you really have to have a disruption of the barriers, because Staphylococcus has to penetrate inside through the skin or the mucosa in order to cause infection.
That could make some sense because the staph A is used to its current niche, and if there's a cut, now the bacteria, enter into this new niche, and I guess the state of that new niche, inside a cut, let's say, is preferential to their proliferation. So they're just able to do it because their conditions are better. It's like someone living in a very harsh climate and they go to a beautiful climate and, they have it easy. They proliferate.
Yeah. So in order to make it even more complex, Staph aureus has a unique ability to go into this dormant stage that is called the persister cell that can avoid it is a metabolically inactive stage where the bacteria cannot be killed with antibiotics. This is not, of course, unique to Staphylococcus aureus, but the percentage of Staphylococcus aureus cells that can transform to this dormant state, it's quite high. In a lot of cases, 100% of the cells of Staph aureus can transform to become persister cells. So that makes the treatment extremely difficult. So the relapse rate is very high. And when, as a clinician, I have to go and give antibiotics to patients with staphylococcal infection, I have to give even longer treatments that are often toxic than in cases with infections with other bacteria. So the problem with Staph aureus is not only the colonization that is very common and very virulent, and it has found a way to colonize us and cause infection through that, but also the ability to stay in this dormant state that it's very, very difficult. None of the existing clinical antibiotics can really kill the staphylococcus when it goes to this persister state.
But that actually could be a very good thing. Instead of a broad spectrum antibiotic, why not target things that make it go into the dormant state or make it come out of the dormant state? So one way would be, if you're infected, give you something that's very targeted that affects those MRSA bacteria specifically and makes them go dormant. Then you have a lot in you, but they're not doing anything to you. I bet you they can't be virulent when they're dormant. They're prepared to keep them in their active state and fight the dormancy and then kill them.
That is a spectacular thought, Rich. Indeed, this is a goal for us. The problem is that this is very difficult. The only target that we can go after when the cells go into this persister state is the membrane of the bacteria, because this is always there. The bacteria cannot really hide. They can hide their, the protein synthesis. They can become dormant, but the membrane is membrane. They need it in order to survive, and we can target that. The problem is that membrane-specific compounds and chemicals are often toxic. So the problem becomes that we have to find something that completely destroys the bacterial membrane of a dormant staphylococcal cell while it does not cause any toxicity to the membrane of the eukaryotic cells and the mammalian cells around it. That is doable. That is something that has been of interest to us, and we feel that we have some unique tools to go after this target. However, this is not an easy target just because of the toxicity.
But again, so when MRSA is in its dormant state, is it virulent, or is it just sitting there sleeping? Is it even consuming material from the outside? Like, how dormant is it? And again, is it virulent at that point?
It is at that point. For example, let's say that it is on a case of osteomyelitis, a bone infection. I start the antibiotics. The staphylococcus, we assume, will go into this dormant state. But when I take away the antibiotic, the MRSA will again express its virulence. So at the moment that I'm giving the antibiotic, at the time that I'm giving the antibiotic, the Staphylococcus is not metabolically active, so it's not causing an active infection. But as soon as I take the antibiotic off, the infection will relapse and will come back. So it's not something that it's not good enough for me to put the MRSA into the dormant state once the infection process has started.
Right, but it may be the easier of the two tasks to do that first. Maybe you have a whole bunch of MRSA in culture, hit them up with thousands of different substances, see which ones put them into the dormant state. They don't have to be necessarily antibiotics, but you've at least partially won the battle if you can do that. If someone's very infected and you could at least put their MRSA into the dormant state, that may buy them days, weeks, months of time for you to do other stuff or for the body to acclimate somehow,
Yeah, no question. No question.
It sounds like you're picking the harder of the two tasks, but the first one I think may be a lot easier,
No question. That's what we do with our antibiotics. We treat staphylococcal infections, unfortunately, all the time. And the way to treat staphylococcal infections is indeed to give a prolonged course of existing antibiotics. So and we are successful. The cost is that of toxicity. The cost is that of unfortunately running the risk of high relapse rate. And high relapse rate means that I don't know who is going to relapse or not, and that forces me to continue antibiotics for longer. But indeed, that is what we do right now, and what we try to do at the same time as a researchers is to try and find a active compound that will target antibiotics while it will target MRSA while it is in this persister form, and destroy it without having to give the antibiotics, the clinical antibiotics, for as long as we do right now.
Do you know what the specific mechanism is by which methicillin kills them when they're not resistant? Because if you figure that out, and then you figure out what has changed inside the Staphylococcus to acquire the resistance to methicillin, that might give you clues on what has changed in it and then what to do about it.
Yeah. Well, the cell wall is the usual target of the beta-lactams. That's the cell wall that changes. Unfortunately, while we are trying to, of course, come up with new antibiotics, and we have had some successes as a field. We have had some progress and some compounds that are active against it. At the same time, unfortunately, resistance also moves on. And Staphylococcus aureus now, there is some resistance to even vancomycin. And there are some strains that are vancomycin intermediate, at least. And also there is some increase in the average MIC, the amount of antibiotic that I need to inhibit the growth of the average Staphylococcus. So it's an arms race in some sort, where we try to advance the science, and unfortunately, as it always happens, bacteria also develop resistance at the same time.
Well, what if the whole thinking needs to be reordered? Forget broad spectrum, because it seems like, at least in part, that it's not just the bacteria you're targeting, but again, what's around it sounds like, again, to me, a critical element of keeping it in check. So if you do a broad spectrum, you're killing everything around it. So that it does two things. It allows the bacteria to become resistant, because it's adapting, but it also kills off the surrounding potential helpers that keep the system in check. So if you didn't go broad spectrum and you thought about it differently, that may be the way to go about it. Go for specifics and never do broad spectrum, because it seems like it's just a matter of time before it's going to fail. So why even go there?
You're absolutely right that we ideally we would like to have narrow antibiotics. And one more reason to try and develop more narrow spectrum antibiotics is the disruption to the microbiome. Because more and more, you use the example of C. diff, more and more when we give broad-spectrum antibiotics, we cause secondary effects by disrupting the microbiome and promoting resistance and promoting healthcare-associated infections that are extremely difficult to treat and have their own mortality. The problem is twofold. The first one is that the problem is diagnostic. So there is a delay between the time that the individual develops an infection and the time that I know the pathogen. It's remarkable to me how we are still waiting for cultures and for bacteria to grow. Clinical microbiology just recently moved to the molecular era as far as bacteriology and mycology. We still, for the most part, most clinical microbiology labs depend on the bacteria to grow, and that can take days, up to 5 days for bacteria to grow, be identified, and then develop an antibiogram. Me as a clinician, not knowing what is going on, I am forced to use broad-spectrum antibiotics because I don't know if the infection is because of MRSA, because of a non-MRSA gram-positive, a gram-negative, or sometimes even a yeast or a fungus. So I find myself in a bind where I'm trying not to disrupt the microbiome. At the same time, the patient, in cases of sepsis, the mortality goes up by the hour. So I see. I don't know the pathogen. So that is the main reason why broad-spectrum antibiotics and empiric use, where we don't know what the heck is going on, and we give 2 or 3, sometimes 4 antimicrobials at the same time, is driving a lot of the resistance, a lot of the toxicity, a lot of the cost associated with the infection. I remember one time, a few years ago, I needed to do whole genome sequencing to some bacteria and for my lab. And at the same time, I was on clinical service. And between patients, I received a call from the sequencing center, and they told me, "Oh, I'm sorry, we need to delay we told you that we're going to have the whole genome sequencing in 5 days. Unfortunately, we're going to have it back to you in 7 days." And I was all upset. At the same time, I found myself on the clinical side to have patients that are septic, that I didn't know what was going on. They were in the intensive care unit. I was bombarding them with 3 or 4 antimicrobials at the same time. And then I would go to the microlab and ask if we have any information on what is causing the infection, and they would say they haven't grown yet. And I would accept that. So the clinical microbiology just recently moved to the area of rapid diagnostics, when, for the most part, rapid molecular identification, even whole genome sequencing, was available to us for quite some time now. So hopefully that will help us have a diagnosis that will allow us to use more and more narrow spectrum.
Yeah, that makes sense. Why was it taking, when you say they were waiting for bacteria to grow, how much did they need, how much of a change did they need to see to say, okay, it's this bacteria, that one? Like, what was the criteria?
The first one is for bacteria to grow. And that, when the bacteria are going to start growing on a petri dish, depends on a number of things. A part of that is depending on the quality of the specimen that you took, the timing, the number of bacteria there. It can be 1 day, it can be 5 days.
But they can't take the specimen and sequence it. They have to let it. They have to sit there and culture it.
Up until now, up until recently, there was no reliable diagnostic tool to do that. Now we are starting to have some tools that have gone through or are going through the FDA approval. So hopefully that will change dramatically and enable the wider use of narrow spectrum antibiotics. And then that hopefully will force or allow the pharmaceutical industry to shift their focus from broad spectrum antibiotics to narrow spectrum antibiotics. Having said that,
I see.
Having said that, we have the other problem with the antibiotics, which is that almost all pharma has moved away from developing antibiotics. And now antibiotic development was left for the initial part to academic labs and to small companies and venture capital. And that is a major problem for our field.
So for all but the absolute sickest people, would it make sense to do shotgun sequencing on them at T0 and then maybe 8 hours later do it again and to see what's changing the fastest? And that might give you an idea of what bacteria are proliferating the fastest and which one needs to be addressed. Would that be possible? And would that be a good solution?
Yeah, there's a number of rapid diagnostic techniques that are coming through, and we're quite excited about those. And indeed, the window that we're looking at that is in real time would be something less than 6 to 8 hours from coming to the healthcare, to the institution, so from presentation. So even if we need to give one dose or two doses of antibiotics, blindly or empirically, how we call it, to use a better term or a euphemism. In any case, when we it is okay. The problem becomes that now with the broad spectrum antibiotics, we give them for 4, 5, or 6 days. And then, by the time that you have the result, the patient is improving. It's a challenge to convince the physician of record to change the antibiotic, because the patient was almost dead. They responded to these antibiotics, and now you're saying, stop all these antibiotics that you're giving and give something that is very narrow. So the sooner we get this information, the earlier in the process we get this information, the less toxicity we're going to give, the more meaningful treatment we are going to provide, and also we're going to be able to address the actual causes of infection. For example, if we know that this is a GI pathogen, then we can say, let's do the CAT scan now and see if there is any illness that we need to address in the GI tract, any surgery that needs to be done. If it's a pathogen that causes endocarditis, let's do the echocardiography earlier on, so we can start and get serious about the underlying diagnosis right from the get-go. So the overall prognosis is going to change, and the overall shift will change. But it's not only the treatment part that needs to change. The treatment part will be based on the diagnostic part.
Yeah, well, I mean, it sounds like since pharmacy companies are not really doing much with antibiotics, and that the whole thought process behind it really doesn't work, at least for very long. I mean, it really needs a rethink, it sounds like. So maybe why not, do it in a very different way? If the support's not there, even if you did find, a new antibiotic, I mean, maybe it's this is really going to pave the way for, targeted antibiotics and not broad spectrum.
Yes, and also incentivize the market. Antibiotics is a unique market where the better the antibiotic, the more we try to avoid using it, because we want to keep it for the people that really need it, for the people that will come with this particular infection. So antibiotic and also the resistance is a risk. Finally, the antibiotics are not priced, are priced relatively low, although they can be real lifesavers. So for all these reasons, pharma has moved away, and the market has to be recalibrated. Otherwise, we are going to have more and more resistant bacteria and no antibiotics to use against them.
Right. I mean, well, this is taking a little bit different direction. Have you or anyone you know profiled the heck out of, MRSA? What does it eat? What does it prefer to eat? What does it not prefer but will still eat? What metabolites does it make? What conditions does it like? pH, temperature, etc. Like, have you profiled its outer membrane, and what does it look like in a normal state versus a dormant state? Like, I would think that if lab profiled the heck out of it, you would definitely find some skews and some uniquenesses and some weaknesses in which to target this thing.
Yeah, well, maybe
don't kill it, starve it to death. I don't know. Maybe
there are a lot of labs that are working really hard on answering every aspect of MRSA and other bacteria. I have to tell you, this is not because of lack of trying, and we do have a reasonable success and progress. The challenge is that, as I told you before, it's a moving target. The challenge is that academia cannot fill the gap that is left by pharma moving out from drug development. I don't have 5 or 10 or 20 or 100 medicinal chemists to work with and to prepare, analogs of compounds that we discover. But we do have a lot of understanding of how the pathogenesis and the pathogenic traits of MRSA make it a different pathogen. We have the same for other very resistant bacteria. And so there is a lot of good biology and molecular biology behind our efforts.
It's all on you, Eleftherios. You got to solve it.
I think that I hope, thankfully not.
I know. I'm just teasing.
Yeah.
Okay, very, very complicated problem. Okay, well, this gives me a lot of insights. I think it'll give listeners a lot of insights into it's not just a black and white picture. It's very, very difficult.
Yeah, I hope that we try to get you into the complexities of the different parts of what we do.
Any wild thoughts about phages? Are there phages that target this particular bacteria in both the dormant and the non-dormant stage? Maybe they would be a way in. Maybe using viruses to again phages really to attack this thing.
Yeah, as you know, there is a increased focus on the development of this tool, and I don't have any insights against MRSA in particular. Nevertheless, there is increased effort now. For the longest time, the development of phages was frowned upon by most Western in most Western countries and industries. But now there is a renewed interest. There is at least one phage therapy that seems to have clinical data behind it. That could be helpful. That could be helpful, especially in decolonizing intestinal tract, focusing treatment. Right now, any novel avenue should be explored.
Yeah, phages may be a beautiful thing because they would be hyper-specific to that bacteria.
Mm-hmm. Mm-hmm. Absolutely.
I guess you got to consider all options,
Yeah. I'm not an expert on phages, but people that are very smart are working on looking into that.
Okay.
There are good presentations, good preliminary thoughts about it.
And are you, this will take us back to the beginning. We're almost done, but is this it's enough, but is this the only bacteria you're working on, or are you working on a whole bunch?
No, we have a different approach in our studies. We go from the question to try and find our way back. So that's how my brain works better. It's I come up with questions. So we do a number of things. We try to use large data to help us find associations with pathogenesis. We try to study the interplay between different pathogens and pathogenic bacteria, non-pathogenic bacteria. So being a clinician, I mostly come up with questions, and then I try to figure out the tools going backwards and trying to answer the question that I found in clinic. And sometimes that can happen with mathematical modeling. Sometimes it can be answered by using large data. often it will happen in the lab, going through wet experiments.
Okay, gotcha. Well, what's the best way for people to get in contact with your lab? Perhaps you, and to suggest collaboration or ask questions or read papers.
Email works fine. Any publications from our group, it has our email, and we encourage feedback, and we depend on collaborators.
Okay, well, very good. Eleftherios, it's been a great call. I really appreciate you coming.
Yeah, Rich, I enjoyed it, and you enjoy the rest of your day.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.